Good afternoon. I am anonymous and I will be helping you get ready for the upcoming microbiology final exam. I know that you are all probably sick of hearing Tischler's voice at 9 in the morning. That's why I decided to do my own thing. I have put a lot of effort into creating this guide and I hope it helps you in some way. Please do understand that you shouldn't only rely on me to get ready for this exam. Use other valuable resources such as the book. Yeah, right. Shaniqua, girl. Ain't nobody got time for that. Jokes aside, I'm going to go over everything from the end of exam 3 up until now. I hope you like it. But before I go any further, let me just say one thing. I don't always make study guides, but when I do, I make sure I make them legit as fuck. Stay in school, friends. Okay. Here we go. Our first lecture is going to be on some microbial diseases. Namely, Legionella, diphtheria, and pertussis. We're gonna start with Legionella. Normally airborne respiratory diseases are propagated. Legionella is one of the only common source airborne diseases. Legionella is very unusual that it is a common source disease. It's not contagious person to person, meaning you have to go to that source where Legionella is to get the disease. The source for Legionella usually is the HVAC, heating, ventilating, air conditioning systems. It frequently goes into cooling towers. It can also be found at hot tubs and fountains. Here's a question. Why all of a sudden in 1970s did they start seeing this disease? Well, more skyscrapers were built, with windows that were lying on cooling towers. Legionella can be treated with antibiotics, because it's bacterial. In 5 to 30% of the cases is fatal. The risk factors are older people who now or formerly smoked, or people with immune suppression and people who are pregnant. How is Legionella detected? You can have an antibody test. Patients who have Legionella pneumonia have antigens for the Legionella in their urine. So clearly a physician will know whether or not you have pneumonia. So they collect urine and see if they suspect Legionella. There's also a water filtration test. Legionella is a gram-negative rod. The Legionella itself is not contagious. Legionella is an intracellular parasite. In humans it infects our macrophages. Out in the wild, it infects protozoa. What happens is that you do not breathe in the Legionella. What you do is you breathe in the amoeba that has the Legionella, and that's how it gets into the lung. That's why it's not contagious. Legionella can colonize on biofilms. Generally they colonize the amoeba, and then the amoeba is then what you get in the shower, or water. Okay, that's all I got on Legionella. Our next microbial disease is diphtheria. Diphtheria is caused by Korean bacteria diphtheria. It's a gram-positive, club-shaped rod. It is an extracellular pathogen. What happens is that the pseudomembrane forms in the back of the throat, and what you wind up with is the toxin that kills the cells that then come and form the membrane. Children die from suffocation, because it cuts off their airways. So the treatment is by antitoxins and antibiotic. However, the antitoxin is more effective. And last but not least, let's talk about good old pertussis. Pertussis is caused by Bordetella pertussis. I don't know why I just sounded Italian there, but let's just go with it. It's also called whooping cough. It's a gram-negative rod. Pertussis is something that lasts for a pretty fucking long time. The first couple of weeks, you have a runny nose, and low-grade fever and occasional coughing. Then you have the second stage which lasts from 1 to 10 weeks, and it's a lot of coughing. Children who die from this, die during this stage. Mainly because they cannot maintain an airway, because they are too exhausted. It's not unusual to break your ribs from this severe coughing. It's very serious. The minimum amount of time for this disease is 3 months, but it can last much longer. This is the P in the DPT shot, my nigga. There are lots of people who opt out of this for whatever reason. Crazy, right? This concludes our lecture. Let's see if you remember any of this shit. Quiz yourself. 1. How is Legionella contracted? 2. Airborne diseases are examples of what kind of epidemics? 3. Legionella is an unusual microbial disease, because it's what? 4. True or false, Legionella is contagious person to person? 5. What is the source for Legionella and where can you expect to find it? 6. True or false, Legionella can be treated with antibiotics? 7. How is Legionella detected? 8. How does a physician know whether or not you have Legionella? 9. True or false, Legionella is gram-positive? 10. What does Legionella target in human bodies? 11. Can Legionella colonize on biofilms? 12. What bacterium is responsible for diphtheria? 13. True or false, diphtheria is gram-negative? 14. What shape is diphtheria? 15. What kind of parasite is diphtheria? 16. How is diphtheria treated, and which treatment is more effective? 17. What bacteria is responsible for pertussis? 18. What's another name for pertussis? 19. True or false, diphtheria is gram-positive? 20. What stage does diphtheria usually become lethal for children? 21. What is the minimum amount of time this disease can last?
Well, hello there. My name is Jennifer and today I'm going to talk about measles, mumps and rubella. I will also briefly touch on bioweapons. So without further ado, let's get started. We have had a large drop in number of cases in measles, mumps and rubella, but it's going back up again. Our first disease we're going to cover is measles. It's caused by the rubella virus. It's a single-stranded, negative, RNA virus. It's enveloped. Symptoms include fever, cough, runny nose, rash. It's highly contagious. Approximately 90% of people who have no immunity and exposed to it, get it. It's not something that doctors see very frequently. There are lots of diseases that were at one time were very common, that aren't common anymore. In 2011, there were cases in the United States in different states associated with communities that are fundamentally religious in some ways. It doesn't matter what religion you are, but there are a lot of fundamental religious people who are against vaccination. Some of the complications of measles include death. The mortality can be as high as 28%. Some people say that it can approach 30%. That's why many people used to die from these childhood diseases. The World Health Organization estimated that in 2011, there were 158,000 deaths from measles. Why are people opposed to getting vaccines? There was a discredited study in England that stated children who get vaccine are more prone to autism. Not true. There are people who advocated no vaccination, and then there are people who feel like if you vaccinate, you're doing something against God's will. Okay? Let's talk about mumps. Mumps is caused by the mumps virus. It's a single-stranded, negative sense, RNA virus. It's enveloped. surprisingly, the symptoms are fever, headache, sort of these general malaise symptoms. But also you get swollen glands. It affects your parotid glands in your neck. If you are an adult man, it's also associated with swelling of the testicular glands. Ouch. That's really, really painful. It's also associated with male sterility. It's thought that the reason George Washington never had children, because he had a documented case of mumps. It's also associated with encephalitis and deafness. Women also get inflammation of mammary glands. Okay, let's move on. Now let's talk about rubella. This disease is also called German measles, or the 3-day measles. It's actually a much milder disease than the other two. It's caused by a single-stranded positive sense RNA virus, the rubella virus. If you remember in class there was a girl who asked Tischler, Dr. Tischler, what do you mean by positive sense and negative sense? This drove Tischler insane, causing her to promptly respond back saying, are you fucking retarded? I've already talked about this shit, anyways, don't be that girl. Positive sense means that their genome can be read directly as mRNA. Negative sense means the RNA has to be manipulated. For example, the A's have to go to U's. The G's have to go to C's before it can be read as mRNA. Symptoms include fever, runny nose, cough and rash. The thing with rubella is not that it hurts the person who has it. The thing with rubella is that the complications are to a developing fetus. That is called congenital rubella syndrome. Complications are heart defects, deafness, eye abnormalities and cognitive difficulties. Depending on when in the development of the fetus the mother gets it, it's up to 50% of the children who are exposed to the utero. Another 20% spontaneous abortions. So one can only guess that whatever is happening to the fetus is so bad that women suffer miscarriages. So it's not necessarily the person who develops the rubella, but the odds of the child who has rubella being exposed to another child whose mother might be pregnant is very high. That was a long-ass sentence. Haha. One of the things when you get pregnant, they always check to see whether or not you have antibodies to rubella, because congenital rubella syndrome is very devastating. If you look at rubella, you can see that the vaccine has worked very well. Because children aren't necessarily getting vaccinated, as you have an uptick in rubella, you see an uptick in congenital rubella syndrome of children who are born. Rubella is not something that is generally fatal. It's a mild disease, but we have to be very vigilant in vaccinating, because we certainly want to prevent the cases of congenital rubella syndrome. Now, let's briefly talk about bioweapons. Bioweapons have been used for a long time. The Tartars lobbed dead bodies of plague victims over a castle wall to try to get their enemies contagious. The anthrax scare in the US didn't sicken many people, but a weapon of terror doesn't need to kill people, it only just needs to scare people. Plague, being an example of a bioweapon, is caused by Yersinia pestis. Yersinia pestis is a gram-negative rod. It's endemic in certain parts of the world. It attacks small mammals, wild rodents. It's vector-borne, and the vector is fleas. There are two different kinds of plagues. There is bubonic plague, which is from a flea bite. The incubation time is 1 to 3 days. Symptoms include high fever, blackening of skin. You can also get swelling, which are called buboes. The swelling occur in your underarms and groin areas. The mortality for bubonic plague if untreated can approach 60%. There is also pneumonic plague. Pneumonic plague is via inhalation. The incubation time is 12 to 24 hours. The symptoms include fever and pneumonia. The mortality if untreated can approach 98 to 100%. What you have to do in terms of treatment is that you have to get treated within 12 to 15 hours of symptoms of onset. That is the end of our lecture. Time for a quiz. 1. MMR stands for what? 2. The viruses that causes measles is known as. 3. What are the symptoms associated with measles? 4. T/F. Measles is highly contagious. 5. The percentage of mortality due to measles is what? 6. Why are people opposed to getting vaccines? 7. What virus causes mumps? 8. Describe the common symptoms of mumps. 9. T/F. Mumps is a positive sense virus. 10. If you are an adult man and you have mumps, what can happen? 11. Name some rare complications of mumps. 12. T/F. The rubeola virus causes rubella. 13. The virus that causes rubella is blank sense and a blank virus. 14. What shape is diphtheria? 15. What is meant by positive sense? 16. What is meant by negative sense? 17. Describe the common symptoms for rubella. What sets rubella apart from mumps and measles? 19. Describe the complications associated with rubella. 20. Why is this vaccination for rubella extremely important? 21. T/F. Plague is caused by a virus. 22. Yersinia pestis is a gram-positive rod. 23. Plague is transmitted by what? 24. The two types of plagues are known as blank and blank. 25. How does one contract pneumonic plague? 26. Describe the symptoms and mortality rate for bubonic plague. 27. How does one contract bubonic plague? 28. Describe the symptoms and mortality rate for pneumonic plague. 29. Within what time frame do you have to get treated upon getting infected with pneumonic plague?
Hello folks, my name is Amy and I'll be filling in for Anonymous. In today's lecture we will cover bioweapons more in depth. Before I continue, let me just say that this lecture on bioweapons is purely for educational purposes. I do not endorse anything covered in this lecture. Also, this is a pretty long lecture, so please bear with me. Okay, great. Let's start, shall we? In the 6th century BC, the Assyrians poisoned a well with ergot, a hallucinogen. It was the darters who threw plagued bodies over a castle wall. In 1931 several League of Nations officials were actually poisoned by a group of people. In 1995, Iraq admitted to having produced 8,400 liters of anthrax, 1,900 of botulinum toxin and 200 liters of aflatoxin. What makes a good bioweapon? Don't make one. There are things that have to happen to make a good bioweapon. What must be important in terms of a bioweapon? It kills other people and not you, so in other words, selective lethality. For example, Napoleon used swamp fever in 1797, it sort of backfired. His troops all got sick. Once you release a bioweapon, you don't have a lot of control over it. Short incubation time would be a good thing, because if you have a long incubation time, how would you really know it was a terror attack? If you're going to have some sort of a terror attack, you want people to be terrorized. You want it to be communicable. You want to have really bad symptoms or no symptoms, like you just want your victim to drop dead. It needs to be an effective pathogen so that it can work its way around your immune system. You want to have an effective mode of delivery. It shouldn't have traceability. You want one that doesn't have an easy antitoxin. It should be toxic. Do you really need to kill people to have a weapon of terror? No. What happened in the fall of 2001? There was an anthrax scare. People were putting anthrax in the mail and it was sent around. Did thousands of people die? No, only 5 people died. What happened? People were going out of their fucking minds. So you don't necessarily need to have a weapon that would be extremely lethal. You want selective lethality. You want short incubation time. Doesn't necessarily have to be communicable. And it doesn't need to have bad symptoms to cause terror. The most important ones are that you want it to be viable, and you have to be able to get it at some place. When you had bacteria in liquid culture, what happened if you saw your culture after a couple of days? They made little tornadoes of snot. If you have little tornadoes of snot, what does that mean? It means that the cells are not viable anymore. What is the snot? The snot is DNA precipitating out. The cells have lysed. If the cells lyse in liquid cultures, then you have a technical problem of being able to store, and a technical problem of being able to deliver it. So unless you're willing to inoculate yourself with Ebola, if you want to spray it on something, it has to be able to survive. Weaponization is a hard thing. It however doesn't take a lot of expertise. Okay, let's discuss plague some more. What did we say was the bacteria responsible for the plague? That's right. Cinnabar. Cinnabar is in urban areas. You get it from a flea bite. Bubonic plague is from tick bites. Mortality/death is between 3 to 5 days. The mortality rate varies from 50 to 75%. Pneumonic plague is through inhalation, and mortality approaches a 100%. You need treatment within 12 to 15 hours. It takes a shorter period of time. The detection is with fluorescent antibodies. The plague has affected a lot of people. During the Great Plague of London, who was so affected by the Great Plague of London, that a whole new branch of science was invented, and what was invented? The answer is Sir Isaac Newton. He invented physics and calculus. The first wave of Black Death came over the trade routes from the Far East. So many people died from it. It was called the Black Plague caused you got blackening of the skin. Why did this medieval doctor wear this creepy-ass outfit? It was to protect against the plague, silly. The purpose of the stick was to keep people away. He had a heavily waxed coat, so that the fleas couldn't get through it to bite him. He had a mask that was filled with dry flowers, so that he would not necessarily smell the stench of death. The plague is endemic in parts of the United States. There is a vaccine for plague. It's for military use, but not very effective. Anthrax is caused by Bacillus anthracis. It's a gram-positive, spore-forming rod. The spores can easily be disseminated. It's not very communicable. It doesn't matter, because it's pretty lethal. You can detect it, but you can have a very effective mode of delivery and its spores can survive. It's normally found in sheep, goats and cattle. So grazing animals pick it up and they inhale it. We know it's endemic in the soil. There are natural cases of anthrax every year. There are cases of anthrax sometimes associated to leather coats, bongo drums and boar bristles. There are 3 different kinds: cutaneous anthrax, which is not very pathogenic; inhalation anthrax, which is the most deadly form; and ingestion anthrax, which is kind of rare and you get it by ingesting contaminated meat. Okay, that's all the time we're going to be spent talking about plague. Let's talk about smallpox now. Smallpox is caused by the variola virus. That's where the name variolation comes from. It's a virus, so it cannot be treated. It gets you sick for a long time. Doesn't necessarily kill people. It disfigures you. It has a mortality of approximately 40%. The illness lasts weeks to months. There is a military vaccine for it. This one is very effective. It can survive and stay viable. It's characterized by a fever. It's very contagious. Symptoms include malaise, vomiting, fever, itchiness and pox. The incubation time is 1 to 3 weeks. People are most contagious before they get sick. Rashes emerge first on the tongue, and then they start becoming pustules. With smallpox, you tend to have them on the extremities, as opposed to chicken pox, where you tend to have them more on the trunk. People are contagious for weeks. Once the scabs have resolved and fallen off, they are no longer contagious. There are 2 different kinds of smallpox. They are known as variola minor and variola major. Variola minor has a mortality of 1% as opposed to variola major, which has a mortality of 40%. The morbidity, or incidence of the disease takes the same amount of time to recover from. Now as usual, time for a quiz. 1. Does a bioweapon need to be necessarily lethal? Why or why not? 2. Why is weaponization a hard thing to do? 3. Name examples in the past where bioweapons have been used. 4. What are the characteristics of a good bioweapon? 5. Where can you expect to find Cine-asbestos? 6. How is Cine-asbestos transmitted, in other words through what vector? 7. What is the more deadly form of the plague, pneumonic or bubonic? 8. How can you detect to see if someone has pneumonic plague or not? 9. Which plague has affected more people throughout the course of history? 10. Why is the plague called Black Death sometimes? 11. Is there a vaccine for plague, and if there is it effective? 12. What microorganism causes anthrax, and describe it? 13. What makes anthrax dangerous? 14. What is anthrax normally found? 15. What are the 3 types of anthrax, and which one is the deadliest? 16. T/F. Smallpox is caused by a bacteria. If false, write the correct answer. 17. What is the mortality rate for smallpox? 18. Is vaccination available for smallpox, and is it effective? 19. Describe the common symptoms of smallpox. 20. What is the main difference between smallpox and chickenpox in terms of where you have the symptoms? 21. What are the 2 different kinds of smallpox, and which one is more deadly?
Anonymous. Yeah, it's me. Did you all miss me? Well, today I plan on talking about foodborne illnesses with you guys. There are 2 major categories of illness. One is infection. With infection, you get colonization and growth, and because of that it will take some time. There is also intoxication. Intoxication means ingestion of a toxin. One of the reasons that we cook our food is to kill bacteria. In 2010, there were 188 reported cases due to bacterial illnesses and outbreaks. It's thought that foodborne illness is very much unreported. The hallmark between an infection and intoxication is fever. Okay, what we're going to talk about first is botulism. Botulism is caused by Clostridium botulinum, a gram-positive, spore-forming, anaerobic rod. It causes a flaccid paralysis. It starts 4 to 72 hours after ingesting the toxin. People are often thought to be suffering from a stroke. Symptoms are difficulty swallowing, difficulty speaking and weakness. The toxin itself is heat-labile. If it's heated for 10 minutes, then the toxin will be denatured. Approximately 30% of the patients die, because generally it's misdiagnosed. There is a test that you can do to see whether or not the toxin is there. It's an immunological test. Next disease is Staphylococcus aureus food poisoning. It's caused by Staph aureus, a gram-positive coccus. It is the second most common reported foodborne illness. However, most staph-related illnesses are not reported. It's associated with uncooked or improperly cooked meat, chicken, salads, pastries, ice cream and eggs. Staph will grow between 8 degrees and 45 degrees Celsius. Most refrigerators are at 5 degrees. Staph is salt-loving. The staph produces an exotoxin, which is incredibly stable. Once that toxin is produced, you get sick. It has a rapid onset of symptoms, 1 to 8 hours. It's pretty fast. Symptoms are cramps, nausea, vomiting, diarrhea. The duration is about 24 hours. The next disease is Bacillus cereus. This is a toxin, an anthracotoxin to be specific. It is very common with rice and starchy foods. It's also found in spices. It produces a toxin, and what happens is that you get nausea, cramps, watery diarrhea. You don't necessarily get vomiting. The incubation is 8 to 16 hours. It lasts 12 to 24 hours. The next disease is Salmonella. It is a gram-negative rod. There are over 2,000 strains known. There are almost 40,000 cases of Salmonella reported annually. Salmonella is associated with improperly cooked poultry and poultry products, as well as frozen foods, sauces or eggs. It's a true infection. Because of the infection, the incubation time is 1 to 3 days. Symptoms include pain, cramps, diarrhea, nausea, vomiting, fever. The duration is anywhere from 2 days to 2 weeks. People sometimes call this the stomach flu. The next disease is Campylobacter jejuni. Campylobacter is caused by a gram-negative rod. It's normal flora in poultry, turkeys and chickens. Between 50 and 100% of animals secrete this. Both Salmonella and Campylobacter are fecal-oral. Now, this doesn't mean you're eating poop. It means things that were in the intestine are getting ingested. There are several cases of Campylobacter a year. It has an incubation time of 2 to 10 days. It's an infection of the small intestine. It has a very nasty exotoxin. It's got to be an exotoxin that's an anthracotoxin. This exotoxin causes inflammation. Symptoms include diarrhea and fever, but no vomiting. Let's talk about another one. Cholera. Cholera is caused by Vibrio cholera, which is a gram-negative rod. It's an infection with the toxin. It has an incubation period of 1 to 3 days. The toxin causes tremendous amount of fluid loss. Symptoms include fever, vomiting. The real problem is diarrhea. Sometimes it's called rice water diarrhea. That's fucking disgusting. You could lose 10 to 15 liters of fluid per day. Death from cholera often comes from electrolyte imbalance. The next disease is shigellosis. It's caused by Shigella, which there are about 4 species of. They are also gram-negative rods. They are generally restricted to human hosts. foods that can cause disease. People who have ingested poultry, milk, dairy products, and salads. Generally things that are handled after cooking. You need very few bacteria to cause a disease. As few as 10 to 100 bacteria. That's what makes this really pathogenic. The incubation is 1 to 7 days. The duration is 4 to 7 days. The symptoms include cramps, pain, fever, nearly vomiting, blood or pus in stools. The next disease is Clostridium perfringens. It's caused by gram-positive, spore-forming anaerobic rods. Clostridium perfringens is famous for causing gas gangrene. It's a common soil bacterium. This happens with proteinaceous foods. It produces a toxin that is an enterotoxin. It's actually called the phage germ, because it's really likely to grow in large portions in a chafing dish. It's an ingestion that actually, you can ingest the toxin or the cells. Either way, the incubation period is 8 to 24 hours. You recover within 24 hours. Symptoms include cramping, pain, watery diarrhea. The next disease is enterohemorrhagic E. coli, also known as killer E. coli. This one is caused by a gram-negative rod. These are E. coli that have a plasmid. They have the Shiga toxin. They have a toxin on a plasmid, which is the O157H7 plasmid. It's associated with beef. It is an infection. It has an incubation period of 3 to 4 days. Symptoms include cramps, bloody diarrhea, nausea, vomiting, fever. There are complications such as kidney failure, especially in small children. And last but not least, let's talk about Listeria monocytogenes. This is a cool-tolerant organism. It's very common in things that you don't cook or reheat. It's very common in cold-cut, deli foods, and raw seafoods. The onset is 2 to 30 days. Symptoms include fever, chills, flu-like symptoms. Infants often vomit and have trouble breathing. In people who have compromised immune systems such as adults, it can be fatal. These people include people who are on chemotherapy or people who have autoimmune disease or women who are pregnant. Okay, that concludes this lecture now. I know there were a lot of information covered today, and I'm sure most of you will put these diseases on your note cards. And that's why I'm not going to do a big quiz for this lecture. I will only ask 3 questions. That's all. Here we go. 1. What is a food infection? 2. What is intoxication? 3. What is the hallmark between an infection and an intoxication?
Hey guys. Amy here again. Today, I'm gonna talk about waterborne illnesses. After listening to this lecture, you probably might not want to swim anytime soon. Here are 2 clicker questions to start us off. 1. True or false? There is more danger from chemical contaminants than there are from microbes? False. There is more danger from microbes than chemical contaminants. 2. By looking at water, you can tell, A. if it's safe, B. if it's unsafe, C. meets standards, or D. if it's wet. The answer is really D, if it's wet. Water could be cloudy and perfectly safe to drink. It could also be crystal clear, but can you see those microbes? No, therefore the only thing you can tell is if it's wet. About half of all waterborne diseases are gastroenteritis. There is also dermatitis and encephalitis. There are all sorts of different waterborne disease outbreaks. What does this map show? It's near bodies of water. The areas that are black are big cities in the United States. They are by the Great Lakes. Do people take vacations to some of these places, such as Florida? Yes, so maybe there is a higher incidence of people in recreational water. In New York there is Niagara Falls, in Massachusetts there is Cape Cod. People go to the lakes in places such as Minnesota, Wisconsin Dells, etc. If you're going to go fishing, are you really in danger of contacting dermatitis this month? No, because if you're ice fishing, you're on top of the water, and you are showing very little skin, so the chances are very slim. Let's talk about Giardia. Giardia is a protozoan parasite. It's eukaryotic because of it being a protozoan parasite. It is very common in rivers and streams. Giardia is endemic in small mammals. Sometimes it's called beaver fever. It's the reason that if you're drinking out of the mountain stream, you're told to either filter your water or add bromine tablets. Giardia doesn't kill you. It's fecal oil. In other words, you're drinking water that a squirrel or a rabbit has pooped in. Yum. It causes some cramping. It's an infection. You don't really get a fever. Maybe diarrhea. Greasy stools and flatulence are the main symptoms. The diagnosis of Giardia is not very good from the stools, because it colonizes sort of the upper intestines and so it doesn't really come out the stools. So the diagnosis is very difficult from poop. What they like to do instead is the string test. What they do is that they have a very long string with a weighted gelatin capsule at the bottom of the string and tape a part of it into your cheeks. Next you swallow the capsule so it goes down and it dissolves in the acids of your stomach, and then you sit there for possibly hours and Giardia colonizes the string. Then what they do is they pull the string back up and then look at it microscopically to see whether there is Giardia there or not. Let's talk about Cryptosporidium. Cryptosporidium is also a protozoan parasite. It's very resistant to chlorination. Usually, when it's summer, you have a lot of water going through water treatment facilities. People use more water in the summer. For example, they might be taking more showers, or watering their lawns. If you have water moving through the water treatment facility very rapidly, you might have less chlorination in the water. For most of us who are healthy, this is not a problem. However, it is fatal to individuals who are immune compromised. This includes people who are taking immune-suppressing drugs, transplants, or people who have HIV. Let's talk about vector-borne diseases. In a public health way, what is the best way to combat a vector-borne disease? The answer is reducing exposure to the vector. For example, yellow fever is mosquito-borne. There was a terrible problem in New Orleans with yellow fever. Even though New Orleans is on the Mississippi River, the Mississippi River is very brackish and salty. It's a tidal river. It's not drinking water. A lot of people in New Orleans had cisterns that captured rainwater. Big, open cistern can be breeding ground for mosquitoes. The simple act of putting screens on top of the cistern can reduce the population of the yellow fever, and indeed it did. Why? The mosquitoes couldn't get through, and they couldn't breathe. So, if you have a vector-borne disease, you want to control the vector. Rabies is a vector-borne disease. What do we do to control rabies? We need to vaccinate our pets for rabies. What is the best way to combat a waterborne disease? The answer is water treatment, or you'll want to do something to make sure people aren't exposed to the water, for example, closing the beach. Okay, let's move on to epidemic typhus. Epidemic typhus is caused by Rickettsia prowazekii, which is a bacteria. The vector is the body louse. If you were a soldier in the Crimean War or a soldier in World War I and you are in the trenches, you don't really have any places to shower, therefore you have to worry about body lice. What happens is you get bitten by the louse, and the louse secretes its feces. If you don't scratch when you get a bite, it's okay. When you do scratch, you rub the feces from the louse into your skin and you're fucked. Symptoms are high fever, rash, lethargy and stupor. Salmonella typhi is fecal, oral, whereas epidemic typhus is vector-borne. The reservoir in the United States for epidemic typhus is the flying squirrel. Lastly, let's talk very briefly about malaria. Malaria is another protozoan parasite. It's mosquito-borne. What you have to do is combat the mosquitoes. The Plasmodium is a protozoan parasite. A lot of people die from malaria in each year. It's endemic in places where you have lots of mosquitoes. Okay. That's all. Now let me quiz you. Are you ready? I think you are, you silly goose. Okay, here we go. 1. True or false, there is more danger from chemical contaminants in water than there are from microbes. 2. About half of all waterborne diseases are what? Gastroenteritis. Oh shit. I just gave you the answer. Oh well. 3. Why are you not in danger of contact dermatitis if you're ice fishing in December? 4. What kind of parasite is Giardia? 5. Where is Giardia found? 6. What's another name for Giardia? 7. Is Giardia fecal oral? 8. True or false? Giardia is an infection. 9. What are symptoms of Giardia? 10. Why is the diagnosis of Giardia not good from stools? 11. What is the string test? 12. Cryptosporidium is what kind of a parasite? 13. If you have water moving rapidly through the water treatment facility, what might happen? 14. What is the best way to combat a vector-borne disease? 15. What kind of a disease is rabies? 16. What is the best way to combat a waterborne disease? 17. What is the vector for epidemic typhus? 18. What is the difference between Salmonella and epidemic typhi? 19. What is the reservoir for epidemic typhus? 20. True/false, malaria is a protozoan parasite. 21. What is the vector for malaria?
'Hello again. It's me, Jenny. Today I will lecture you on wastewater treatment. Honestly, it's a boring one, so if you need to take a break or something like that, do so now. Otherwise, get ready to be bored to death. Alright, let's start. Water purification is what happens to water before it comes out of the tap. Water contamination is very dangerous. Microbial contaminants are far more dangerous than chemical contaminants. The goal of wastewater treatment is to have safe drinking water. Untreated water is aerated. It might be softened depending on where it comes from. Softening involves removing some minerals, calcium and magnesium, which aren't necessarily dangerous, but certainly build up inside pipes. Then removing any turbidity, any bad odors, or tastes, and then finally disinfecting it. So, what is it that we really need to remove? We need to remove the things that are suspended. That's called total suspended solids. They make the water cloudy. It doesn't matter what they are, and it could be things that are organic or inorganic. When you add some chemicals to it, what happens is that it makes the small particles bigger. That's called flocculating, so that they can settle out. You can lower the total suspended solids, either by settling, which is what's done more frequently, or by filtration, and you might be filtering at the faucet or something like that. But this is municipal water treatment. You might add chemicals that then help to remove the turbidity via settling, and the final step then is chlorination. Chlorination in the US is what is used because we take our water for long periods of time, long distances. The water that comes originally from Lake Michigan is what comes out of our tap. In Europe, they generally add ozone, but they are not transporting their water so very far. You can also irradiate the water. In the US, we use chlorine, because it stays with the water. Once they started filtering their water, the number of typhoid cases went down, and chlorinating the water also helped. How do we know if water is clean? We really don't. We can test all of the water. We do in statistical approximation. It's called MPN, or most probable number. It's a conservative estimate of an indicator organism. And the organism that we use is called the coliform bacteria. It's like E. coli, but it could also be a close relative. It doesn't require identification of a pathogen. We know how many different pathogens cause diarrheal diseases that are from the fecal-oral route, and so it would be horrible to ask, is it this, is it that, or is it something else? But what the MPN asks is that might there be a pathogen in the water, and if there is, it doesn't matter. If there is some sort of contamination, then we're going to be very conservative and we're going to take precautions. However, it's the growth-based test, because it's not asking for something specifically. This figure shows that you can take a water sample, you dilute it in a lactose broth, and you can see that there are erm tubes, and the gas collects in these tubes. So what you're looking for is what is the lowest number, or the lowest dilution that you have, acid and a gas that is produced in the lactose broth. Then, you can go further and confirm and complete the test, but it's that 24-hour growth presence of gas in a lactose broth that gives you that. And that's why with Lake Michigan, they closed the beach based on what they found the day before. Why do you use lactose broth? Lactose is a sugar that is in milk. Mammals make milk. If you use something else like glucose or sucrose, you would get tons of bacteria, but the bacteria that go through the guts of mammals, which are the bacteria that might cause disease in mammals, are those bacteria that might use lactose. Once you have the dilution out after a positive test at 24 hours, you might want to close the beach. And then, you'd continue on to make sure that you really did have coliform bacteria. The MPN is a very conservative estimate, and it looks for things like E. coli. E. coli is very long-lived. It's present in larger numbers in a fecal sample, showing fecal contamination, when there might not be any pathogen. It says, pathogens might be there, not that they are necessarily there. We do this to make sure that our water is biologically safe. This is what we do with municipal water. Water from the well is not the municipality's responsibility. Let's talk about wastewater treatment. Wastewater treatment is what happens to the water once it goes down the drain. The goal of wastewater treatment is to have environmentally safe disposal of the wastewater. What's in the majority of wastewater? Mostly water, but there is organic matter. That organic matter acts as food for any microbes that are going to be in the receiving body of water. When the microbes metabolize the organic material, they use oxygen, and it doesn't matter what the organic material is, because you're going to find a microbe in the environment that's going to be able to use that material. When you have too many microbes using up all the oxygen, you wind up with fishies getting killed. That's so sad that it makes me want to cry. Sniffle sniffle. Let us talk about biological oxygen demand, or B.O.D. It doesn't really matter what the organic matter is. Just that it's organics that microbes can use to grow and use up the oxygen. So the microbe grow. They use up the oxygen, and the more organic matter there is, the more oxygen is used. So, what you want to do is to dispose of the water in an environmentally safe way. So you need to reduce, not only the total suspended solids, but also the biological oxygen demand. So we have sewage treatment plants. Commonly, there are suspended solids, biodegradable organic compounds. You also want to remove pathogenic microorganisms that happens as part of the sewage treatment process. You need to make sure that you get rid of pollutants that are toxic and heavy metals. Wastewater treatment is designed to do that. So there are 3 steps: primary, secondary and tertiary. Secondary steps are almost exclusively biological. Primary is a physical settling. Tertiary can be either chemical or biological. Let's look at figure 43.4. Look at where the water comes into this sewage treatment plant. The little pink parts are the primary clarifiers. So what happens is the water comes in to the sewage treatment plant, and it is screened, and it's filtered, and there's a lot of engineering that goes into this. You can see that there is a series of screens. It's called a grid chamber, where you get settling. Barbie dolls toss down the toilet, eggshells ground up in garbage disposals, all of those go into the sewage treatment plant. They get stopped by the screens in the case of the Barbie dolls. In terms of the eggshells, they go to the grid chamber and they settle out. Primary is physical settling. You're physically settling some of the contaminants in sewage or wastewater. By doing that, you remove 40 to 60% of what is in the wastewater. Figure 43.6 shows the different ways that you can have secondary treatment. The first on the left is activated sludge, where you have active aeration to get the organic matter eaten by the microbes as fast as possible. The one on the right is called trickling filter, where you have the sewage sprayed, and they are sprayed by little levers, therefore the water trickles through. You have bacteria and biofilms and slime growing that eat the carbon, eat the organic material as it passes through. The problem with the trickling filter is that places like Chicago really wouldn't do really well with a trickling filter. Those tend to be in more temperate places. In this picture, on the right we have activated sludge, on the left we have the trickling filter. If you have extra time, you have something a little bit longer, but it's still an activated sludge system. How fast things go through depends on how fast the water is going through. After the organic matter has been removed, in other words, it's being transformed into microbial cell mass, it then gets digested. It gets settled, and it goes off to a digester. There are several different steps like we've seen in the rumen of a cow and the hindgut of a termite. Wherever you have fermentation, you have acetogenic or acetate made. And you also have methane or natural gas made. These take place in an anaerobic digester. You have water coming in from the primary treatment. In the aeration tank you can find activated sludge. What you're doing is you're turning the organic material into biomass. You're then settling it, and then what you're doing is you're reducing the volume, because what you have is cells that were made aerobically. There is not as much energy to digest things anaerobically, and so you have a reduction in biomass. Here's a schematic of an anaerobic sludge digester. You have actively digesting sludge. That's the biomass from the aerobic step being used. By the time you get to this point, you have reduced the pathogens. They're no longer there. They are either dead or food for the anaerobic microbes. Then the stabilized sludge can be used for its nutrient value. Another schematic of anaerobic sludge digestion. This is very much like what goes on in the cow's rumen. You have complex polymers, which were the microbes that ate whatever was in the wastewater. The microbes that were made aerobically die. They are hydrolyzed, fermented, and then methane is made. Figure 43.8 shows that if you do not have a municipal wastewater system, what you have is the septic system. This is common in rural areas. You have a little tank. The bacteria settle out, and you wind up with some sludge. Septic tanks eventually fill with sludge, and then the water goes in the septic field, and you need to have enough land for a septic field. Okay, that's all there is to this lecture. Thank God. At least you didn't have to listen to Tischler's voice. Now, I don't know about you, but I'm tired of talking. I went over a lot of information, so I hope you're ready for a quiz. Let's get started. 1. What is the goal of wastewater treatment? 2. What does TSS stand for and what is it? 3. What is flocculating? 4. Name 2 ways you can lower the TSS. 5. What is the final step in water treatment? 6. What does MPN stand for and what is it? 7. What organism is used for MPN? 8. Why do we use lactose broth? 9. Name the 3 steps to wastewater treatment. 10. What is each step for? 11. What happens when you have too many microbes using up all the oxygen? 12. What are the 2 types of secondary treatment that you can have? 13. What is the problem with trickling filter? 14. What is the purpose of secondary treatment? 15. How is secondary treatment of wastewater similar to what we have seen in ruins of cows and hind guts of termites?
Hey there. It's me, Anonymous, again. Congratulations. You have just finished listening to all the lectures. I hope you all thoroughly enjoyed it. Feel free to listen to it as many times as you want. Over the next few minutes, I will show you questions from the final exam from a couple of years ago. Some of them I have answers to and some of them I don't. Since Tishel is aware that the old final is circulating around, I doubt that she will use many questions off of it. Nonetheless, it's still a good tool to study off of. Before I continue, let me say one last thing. If you think this video helped you in any way, please be a kind-hearted soul and consider donating. Remember, I do this all for free in my spare time, and I'm a starving college student. I will provide a link in the description box. Once again, thanks for listening. Have a fan-fucking-tastic day. I'm out.
Sing along a song that's so sensual. Bring along a sip to make it oh so sexual. Verbally that is making love to the music means vibing to the beat at night. With the whole city fast asleep. Out cold true words seem to rise to the lips. Take hold of a poet of me most powerfully. I feel free when the world doesn't know it to me. It's so hard to find a gig that lives up to the billing. Trying to find a reason to work out, willing I admit my thinking is wishful. Like a star upon a child gazing up to the ceiling. How far do we have to stretch the truth to fit the lifestyle borrowed and overdue? We can take it all back to the register and start all over from the can of stuff. Let's break it all down into pieces of bright moments that pass by like a meteorite. Throw on your favorite reel, that's good to go. On the analog player, watch the people glow. Sit back with the breeze and let the memories flow. Comedy, tragedy, all the highs and lows. It's funny how the music puts times in perspective. How the soundtrack to your life is perfected. Whenever you are feeling blue, keep walking and we can't get far. Wherever you are, it's funny how the music puts times in perspective. How the soundtrack to your life is perfected. Whenever you are feeling blue, keep walking and we can't get far. Wherever you are. Like a move that I can't predict, like a look that I can't resist. The tingling feeling was so mutual. The lingering appeal was so unusual. Herbally, what is medicine to a lone soul can become poisonous. Some with the whole body fast asleep. Out cold, true vision seem to come to the eye. Take hold of a prophet of me most visibly. I see clear when the world doesn't show it to me. It's so hard to make sense in the cycle of billing. Trying to find a reason to quit and make a killing. I admit, our dealing is painful. Like a star upon a child staring down from the ceiling. How far do we have to stretch the picture before pixelating the human texture? We can take it all back to the register and start all over from the can of stuff and save it all up for an ultimate prize. Homecoming gathering with the big surprise. Throw on your favorite record that's good to go on the analog table and it's something to blow. Sit back with these and hear the MC flow. Hi-hat, kick drum, all the highs and lows. It's funny how the music puts times in perspective. How the soundtrack